Finally, the organ system you've been waiting an entire academic year for. As usual, we'll focus on anatomy in the laboratory and physiology in our lecture. With the reproductive system, we see the endocrine system at play. In other words, the hormones control the cycles. There is similarity in the hormonal pathways of males and females. Organs of the reproductive system are divided into two categories. Gonads, the primary sex organs because they produce both the gamete, a sperm or oocyte, and reproductive hormones, testosterone, estrogen, or progesterone. Everything else is helpful and considered an accessory organ. These include ducts to carry the gametes, glands for non-hormone secretions, and the genitalia. We are going to look at physiology of males first. In general, the hormonal cycle controlling gamete formation in males starts at puberty and is fairly constant. We will see traditional negative feedback preventing large swings in the hormones. As stated earlier, the testes are the primary sex organ because they are the gonad producing the gametes and specific hormones. The gonad itself has two outer layers, a tunica vaginalis and a tunica albuginea, the latter being deep. The tunica albuginea invaginates to form septa. Within each area is a long coiled up tube called a seminiferous tubule. It is in these tubules that sperm formation will take place. Once these sperm are made, they are moved through a duct system consisting of a rete testes, then into the epididymis. Sperm finish maturing and are stored in the epididymis. Upon ejaculation, the sperm are moved through the vas deferens into the body cavity. There are blood vessels, nerves, muscles, and fascia traveling with the vas deferens. That entire group is collectively called the spermatic cord. Sperm and gland secretions are mixed together through an ejaculatory duct to form the semen, which will move through the urethra. You'll see all these structures in the laboratory. Now, sperm formation requires different cell types that are inside the seminiferous tubule. Between the tubules, we will find interstitial cells which secrete testosterone. Males have three sets of accessory glands. Seminal vesicles are posterior to the bladder, and their secretions are mixed with the sperm in an ejaculatory duct. 1. Seminal vesicles secrete an alkaline mixture of fructose for energy, hormones called prostaglandins, and coagulating enzymes. 2. Prostate glands are located inferior to the bladder and are responsible for the milky appearance of semen. Their secretions are slightly acidic and contain citrate for energy and enzymes for sperm activation. And 3. Bulbourethral glands are in a different location in the muscle wall of the base of the penis. Their secretions are released before the semen and are responsible for neutralizing the environment of the urethra. It turns out that sperm are extremely sensitive to pH. And as we learned in the previous organ system, the pH of urine can vary widely. So these glands release a secretion that neutralizes any base or acid that may have been in the urine. This maximizes the survival rate of sperm through the urethra. Semen is, therefore, a mixture of a lot of things. Functionally, Semen is the transport medium and nutrient source for sperm, which are living cells. There are components that provide protection for the sperm and enzymes that activate them, needed for fertilization of the oocyte. Several prostaglandins are included from seminal vesicles. These help make the mucus of the female cervix thinner so that more sperm can get past. Other prostaglandins stimulate reverse peristalsis in the uterus, which helps more sperm get to the oocyte. And others help the sperm be better swimmers. Other inclusions in semen are relaxin, which again makes sperm better swimmers, seminal plasmin, sort of like an antibiotic to prevent bacterial degradation of the sperm, enzymes to help the semen coagulate in the female tract so that it stays in the female tract longer, and lytic enzymes to break the clots after a period of time. As you can see, seminal components are really trying to get someone pregnant. Sexual reproduction allows for the mixing of genetic material between different individuals. The beauty of this is that it dramatically increases genetic diversity in that species. In general, a gamete from each parent unites to form a zygote, a genetically new individual. Now, your regular body cells are called somatic cells as a collective group. Somatic cells contain 46 total chromosomes, but those chromosomes exist in pairs. The two chromosomes in a pair code for the same information or genes, and they are identified and numbered. There are 22 somatic pairs and one sex pair. Now, if you took a gamete with 46 chromosomes and merged that with another gamete with 46 chromosomes, you'd end up with a zygote that has 92 chromosomes. That won't work. So we need to solve this problem. We need to make gametes that have only 23 chromosomes, but still have one chromosome from each pair, so that the information is still basically complete. Meiosis is our solution. Meiosis is two cell divisions back to back that takes one diploid parent cell. Diploid means chromosomes are in pairs, and results in 4 haploid progeny cells. Haploid means chromosomes are alone and not in pairs. Through meiosis, we can take a diploid cell and create haploid gametes. Again, meiosis is two divisions back to back. In the first division, one diploid parent cell divides into 2 haploid cells. Then each haploid cell divides into their haploid progeny cells. If you've forgotten meiosis from general biology, now is a good time to stop this lecture and use the internet to find a short review video. We are under the assumption here that you know what meiosis is and exactly how it works. In males, the cells of the seminiferous tubule that will give rise to the final gamete are called Spermatogonia. These are diploid and become functional at puberty. Spermatogonia divide by mitosis to create 2 diploid progeny cells. This means that males constantly replace their spermatogonia. The 2 progeny cells are called A and B cells. The A cell stays a spermatogonium and continues to divide by mitosis. However, the B cell will undergo spermatogenesis to become a gamete. We will see the specific stages listed here on the next slide. But a B cell becomes a primary spermatocyte, which divides by mitosis to become secondary spermatocytes, which finish meiosis to eventually become spermatid. There's a lot on this diagram, so let's break it down here. This is a diagram of the wall of a seminiferous tubule. Look on the far right, and you'll see that the tubule wall is divided into a basal compartment and an adluminal compartment. Now focus on the top of the diagram at the very edge of the tubule wall. You'll see that there are two cell types located at the edge of the basal compartment. The regular-looking cell is the diploid Spermatogonia. Every now and then, you'll see a weird-looking cell called sustentacular cells. More about those later. Let's follow the Spermatogonium's fate. First, it divides by mitosis to create an A and B cell. The A cell stays in place and keeps doing the same thing. But the B cell begins to migrate toward the adluminal compartment. At the same time, it starts to undergo physical and genetic changes and becomes a committed cell. Once it's committed, we will call it a primary spermatocyte. You'll see in the lab that they look a little different. Primary spermatocytes are diploid, and when ready, will complete the first meiotic division. The progeny cells will be haploid and are called secondary spermatocytes. Note how the secondary spermatocytes are continuing to move through the adluminal compartment. Secondary spermatocytes will then complete the second meiotic division to become early spermatids, still haploid. Early spermatids are still not gametes. Note how they look like a traditional cell. These cells have to now undergo a specific process called spermiogenesis. During spermiogenesis, they will lose their extra cytoplasm and grow the structures needed to be fully functional sperm. Once that happens, the fully formed spermatozoa will be released into the lumen, where they will travel through the rete testes to the epididymis for maturation and storage. A diagram of spermiogenesis. The spermatid loses its extra cytoplasm and grows several structures. Flagella for motility, a midpiece which houses mitochondria for ATP production, the nucleus containing the genetic material, and an acrosome which contains enzymes needed for fertilization. Spermatogenesis requires more than just the spermatogonia, including sustentacular and interstitial cells. Remember those weird-looking sustentacular cells inside the seminiferous tubule? They have a lot of functions. First, they help form a barrier between the immune system and the forming sperm. Remember, sperm formation begins after the immune system has become immunocompetent. The developing sperm would be seen as foreign by the immune system. So the sustentacular cells form tight junctions and block access by the immune system. These cells also provide nutrients to the developing gametes and move them through the wall of the tubule toward the lumen. They are phagocytic and will remove the extra cytoplasm and dead gametes. Lastly, they secrete an important protein called androgen binding protein. ABP will bind testosterone so that it stays available in the wall of the tubule to drive these processes. Interstitial cells are found between seminiferous tubules, hence their name. Their function is straightforward. They produce testosterone. Testosterone will diffuse into the tubule, then be held in place by ABP. A diagram of all the hormones here. The endocrine control of all this excitement is called the brain-testicular axis. Let's start at the top, literally. The hypothalamus ultimately controls gamete production. It secretes gonadotropic releasing hormones, which travel through a local capillary bed to the anterior pituitary. In response, cells of the anterior pituitary secrete two hormones, luteinizing hormone LH and follicle stimulating hormone FSH. Their names will make sense when we see them in females. Now, FSH and LH are both released into the blood supply. They will travel to their target cells in the testes. LH will stimulate testosterone production from interstitial cells. But FSH affects sustentacular cells, stimulating them to secrete ABP, as well as a hormone called inhibin. The dashed lines on this diagram show us the negative feedback loops, and there are many in males. First, testosterone provides negative feedback on both the anterior pituitary and hypothalamus. So elevated testosterone levels will shut down testosterone production. We can also see that inhibin provides negative feedback at the anterior pituitary. Again, these negative feedback loops keep testosterone levels fairly consistent in males. And since testosterone drives spermatogenesis, that means sperm formation is relatively constant in males. As males age, they will experience some decrease, but they do not have a complete shutdown like females do. Here, I've listed the specific hormones for you to see more clearly. GnRH is released from hypothalamus and stimulates FSH and LH production from the anterior pituitary. In reality, there is a specific GnRH for each hormone. FSH is released from the anterior pituitary and stimulates sustentacular cell production of ABP and inhibin. LH is also released from the anterior pituitary, but it stimulates interstitial cells and testosterone production. Not only does testosterone drive spermatogenesis, but it has secondary effects throughout the body. Testosterone does affect the brain and controls the libido or sex drive. It stimulates pubic, axillary, and facial hair growth. It's anabolic to skeletal muscles, masculinizes the skeleton, and makes the skin thicker. Now we are ready to look at female reproductive physiology. There are some similarities to males, as the hormonal axis does utilize GnRH, then FSH and LH from the same glands. However, the story pretty much goes downhill from there, as females are cyclic, have multiple overlapping cycles, and don't alternate between negative and positive feedback loops. Anatomically speaking, the ovary is the gonad as it produces the gamete or ova, and the hormones estrogen and progesterone. Accessory organs are the duct system, uterine tubes, copulatory organ vagina, and a gestational organ, the uterus. Whereas testes are pretty organized with septa and tubules, ovaries are quite different. The outer tunica albuginea is still there. But the outermost layer of cells in the ovary itself is called a germinal epithelium. That tissue will be broken through, so it has to do mitosis easily to heal itself about every 28-ish days. Do not use that number as a birth control method. Now, within the ovary, we find follicles. You can think of a follicle as an egg house. The inside of the follicle contains the developing oocyte or ova, and that oocyte is surrounded by functional cells. It's almost like a math formula. Follicle equals oocyte plus outer cells. Follicles are usually in the cortex region of the ovary. Do all the follicles look alike? Nope. Follicles will go through many developmental stages. They'll begin as tiny ones, the blue circle, grow and explode during ovulation, the red circle, then change again into a glandular structure, the green circle. Again, a follicle is a cell-based structure inside the ovary that contains an oocyte. Interestingly, these form before the end of the first trimester of that female's gestational life. For example, before my mom was 3 months pregnant with me, my ovaries were formed and contained fully made follicles. Females are born with follicles that are in a form of suspended animation, not metabolically active at all. That first follicle is made of a single layer of follicle cells. Once puberty begins, in each monthly cycle, a few of these follicles will become metabolically active. At that point, they are said to be made of granulosa cells. It's the same cell population, but when they're asleep, we'll call them follicle and cells, and when they're awake and doing their thing, we'll call them granulosa cells. You'll see examples of both in the lab. These are the 3 cycles we will be looking at. Ovarian events that have to do with follicles and oocytes. Menstrual events that have to do with the uterus, and hormonal events that control everything. This flowchart shows us the developmental sequence of the ovarian follicles. In red italics, above each one is the ploidy, diploid or haploid, of the oocyte inside. We are born with primordial follicles, but don't start to develop primary follicles until puberty. At that point, the sequence is primary follicles become secondary follicles, become Graafian or vesicular follicles, reach ovulation. A diagram of oogenesis. Here, we are focusing on the oocyte inside the follicles and how they develop into a functioning gamete. Oocytes are made by their precursor cell called a diploid oogonium. The oogonium undergoes mitosis and then becomes a committed primary oocyte. That primary oocyte begins the first meiotic division. However, at prophase 1, it stops the process and goes into a suspended state where it waits until puberty. Mind you, this process takes place in utero, while this female is still inside their mother's womb before the end of the first trimester of pregnancy. Once puberty happens, hormones will act on a few follicles with each cycle. Upon hormonal stimulation, the follicle will grow, and the oocyte inside will change as well. A specific hormonal event will cause the primary oocyte to finish meiosis and to become a secondary oocyte. Now, the division of cytoplasm is not balanced. One cell will get most of the cytoplasm and stay an active cell. That is the secondary oocyte. But the other cell is tiny with very little cytoplasm. It is called a polar body and is not a gamete at all. The oocyte is released during ovulation into the ducts. If the secondary oocyte is fertilized, it will then finish the second meiotic division to become a fully functioning ova that pretty much immediately becomes a zygote. If fertilization doesn't occur, the secondary oocyte will actually die without having finished that last division. We still see the similarity in the pattern. A diploid or ogonium becomes a diploid primary oocyte. That finishes meiosis and becomes a secondary oocyte. That finishes the second division to become the gamete. Differences are one oogonium becomes one gamete. We can divide the events occurring in the ovary that pertain to the oocyte and the follicle into 3 phases. The follicular phase, days 1 through 14, noted by increasing estrogen and growth of the follicle from primordial to primary to secondary. Ovulation around day 14, noted by a surge in LH hormone. This triggers final maturation of that oocyte. Its follicle explodes or ruptures, and the oocyte is released. The luteal phase, days 14 through 28, noted by formation of a corpus luteum producing progesterone. This hormone prepares the uterus for pregnancy. If pregnancy does not occur, the corpus luteum generates to a corpus albicans. This diagram shows the same sequence of events, only we'll add in the follicles. First, quick review of the oocyte. The oogonium becomes a primary oocyte that is still diploid before birth. That primary oocyte stays in its state until hours before ovulation, where it finishes the first division and it becomes a haploid secondary oocyte. The secondary oocyte is released from the ovary and only completes the second division to become an ovum if it is fertilized. Now let's look at the follicles. Follicles have several developmental stages. Remember, think of the follicle as an oocyte house. That house will change throughout the month cycle 3 times. The first phase, follicular phase, is usually the first 2 weeks of any given monthly cycle. Again, it's called the follicular phase because we see maturation of that follicle. The primordial follicle becomes metabolically active, and the follicle cells wake up and start dividing. Now we call them granulosa cells. Once we see layers in the wall of the follicle, it's now a primary follicle. That will continue to grow under hormonal control, getting bigger and bigger. At one point, the granulosa cells will start to separate from each other, and we will see a fluid-filled space or antrum appearing. We now have a secondary follicle, which now starts secreting estrogen. This continues to grow and secretes estrogen in a positive feedback manner until the follicle is huge, the antrum is gigantic, and the amount of estrogen produced is extraordinary. Then magically, the follicle will switch estrogen production to negative feedback and the estrogen levels crash. This will trigger a hormonal change. The secondary follicle has become a Graafian follicle. Now, this hormonal change places us in the secondary phase, ovulation. This short, 24-hour-ish event finds the follicle, the vesicular or Graafian follicle, suddenly filling with extra fluid and exploding. The oocyte inside will complete the division to become a secondary oocyte. The secondary oocyte, with a thin layer of granulosious cells around it, a corona radiata, is expelled from the ovary. Once again, the hormones will change. The last hormonal change triggers the third and final ovarian phase called the luteal phase. Within a couple of days of ovulation, the granulosa cells left behind in the ovary will divide and become something called a corpus luteum. The corpus luteum now secretes progesterone and some estrogen to prepare the body for pregnancy. The corpus luteum lives about 14 days. If pregnancy does not occur, it disintegrates into a corpus albicans and dies. So estrogen and progesterone levels fall once again. If pregnancy does occur, the corpus luteum stays alive and will produce the hormones throughout the first trimester of pregnancy. Pretty amazing stuff. By the way, do not use these numbers as a birth control method. Only the luteal phase is predictable. The follicular phase varies each month, and you don't know when ovulation occurred without testing for it until it's over. And those lovely sperm can live up to 5 days in the female tract. Counting days is the rhythm method, and it does not work. Natural family planning is something else, and that can work. Again, the luteal phase is named after the corpus luteum. It lasts about 14 days, and the fate of the corpus luteum depends on whether pregnancy has occurred. We've looked at the ovarian events. Now let's look at the hormones that control all of that. If you start near the hypothalamus, you'll see familiar hormones. The hypothalamus secretes gonatotropic releasing hormone, which stimulates FSH and LH production from the anterior pituitary. Unlike males, however, this is not simultaneous. More FSH is produced than LH in the follicular phase. That FSH is what stimulates growth of the follicle, hence its name, follicle stimulating hormone. The follicle grows and grows to become a secondary, which starts secreting a lot of estrogen. At first, estrogen is positive feedback, so we get more GnRH, more FSH, and more estrogen. As mentioned before, when estrogen reaches peak, that will switch to negative feedback and estrogen levels will fall. The crash in estrogen levels triggers a quick surge of LH hormone. It is the LH surge that triggers ovulation and the formation of the corpus luteum. During the luteal phase, estrogen, progesterone, and even inhibin are negative feedback on the hypothalamus, preventing any more GnRH during this cycle. Overviews of these hormones. Let's review again. On day 1 of any monthly cycle, all hormone levels are low. The hypothalamus secretes GnRH, which stimulates FSH production from the anterior pituitary. FSH levels rise and stimulate growth of the follicle and its positive feedback. Once a secondary follicle is formed, estrogen is produced, and is positive feedback. FSH, follicle growth, and estrogen levels now rise quickly. When levels peak, estrogen becomes negative feedback at the hypothalamus and levels crash. This triggers a surge of LH hormone, which triggers final maturation of the primary oocyte into a secondary oocyte and ovulation. The oocyte, with its corona radiata, are released, and the remaining granulosa cells develop into a corpus luteum. That corpus luteum secretes progesterone and some estrogen for the next 2 weeks. During the luteal phase, rising progesterone prepares the uterus for pregnancy. Again, during the luteal phase, rising progesterone prepares the uterus for pregnancy. More about that in a bit. And remember, if pregnancy does not occur, the corpus luteum degenerates into a corpus albicans, and the hormones will fall again. We've looked at the events of the follicles and the oocyte and the ovary, as well as the hormones that control those events. Now let's see how the same hormones control the uterus. Uterine events can also be divided into 3 general phases, assuming that day 1 is the first day of the menstrual cycle. Days 1 through 5 are the menstrual phase, where the stratum functionalis of the endometrium sloughs off and falls out of the tract. In lay terms, this is the period. It is not blood, but a highly vascular tissue that is expelled. All hormone levels are low at this point. Days 6 through 14 are the proliferative phase. During this time, GnRH and FSH levels are rising, as well as estrogen eventually. The rising estrogen starts the rebuilding of the stratum functionalis of the endometrium. Days 15 through 28 are the secretory phase. Ovulation has occurred, and the corpus luteum is secreting progesterone and some estrogen. Those hormones dramatically affect the uterus, with significant rebuilding of the stratum functionalis taking place, as well as secretion. For example, glycogen is made to serve as an energy source for the developing zygote, as well as paracrines that will assist with implantation and maintaining the pregnancy. Progesterone is the primary hormone acting here, with estrogen playing a secondary role. Should pregnancy not occur, remember the corpus luteum will degenerate. On about day 26, progesterone levels start to crash. Progesterone was stimulating and maintaining the stratum functionalis. So this crash means there's no longer a hormonal mechanism to keep the stratum functionalis in place. The arteries within it kink and the tissue sloughs off. This will be day 1 of the next cycle. A great yet complex diagram. This diagram puts everything together. At the top, we have the 3 phases in the ovary. In the middle are the ebbs and flows of FSH, LH, estrogen, and progesterone. And at the bottom, we see the 3 phases of the uterine cycle. You can even see a little anatomy of the endometrium. See how there are permanent vessels called spiral arteries and veins in the stratum functionalis? Each month, under progesterone stimulation, the ends of those vessels will grow into the newly forming stratum functionalis. These are the vessels that progesterone holds open to maintain the stratum functionalis in preparation for pregnancy. Spend some time with this diagram. Be able to explain the rising and falling of hormones in any uterine or ovarian phase. also be able to explain the uterine or ovarian results of those hormonal changes. Yeah, this is pretty complex. It is not your imagination, and there's a lot going on here. Estrogens and progesterones have secondary effects in the body. Estrogen helps drive oogenesis. No ripening of follicle or oocyte without it, and growth of reproductive hormones during childhood. Estrogen also stimulates motility and mucus secretions of those same organs. Progesterone tends to prepare the body for pregnancy and keeps the spiral arteries open, leading from the stratum basalis into the stratum functionalis and operating during the secretory phase. Other estrogen effects in the female: growth of breasts and the myometrium of the uterus, feminizing the skeleton, stimulating axillary and pubic hair growth, and favoring subcutaneous fat deposition in hips and breasts. Lastly, the sexual response for delivering the gametes is similar in males and females. This makes sense as we both develop embryonically along similar paths. The parasympathetic nervous system is responsible for moving blood into genitalia, including the penis. Ejaculation and orgasm, however, are controlled by the sympathetic nervous system. There's a mnemonic for remembering this. It's point and shoot. Erection is parasympathetic. Ejaculation is sympathetic. Yeah, you're right. We only talked about physiology. We did not talk about sex. That's a completely different class.